Welcome to this month's EMBL ABR, Australian Biocommons and ARDC webinar, which will be about multi-omics statistical integration with mixOmics. My name's Christina Hall and I'm from Melbourne Bioinformatics, which is a node of EMBL ABR, and I'm your host for today. My colleague, Susanna Bacon from ARDC, is behind the scenes co-hosting the webinar. Today, we're very pleased to have Dr. Kim-Anh Lê Cao speaking to us. Kim-Anh is a senior lecturer at the University of Melbourne in the School of Mathematics and Statistics. Kim-Anh is fascinated with numbers and how innovative statistics can influence the world. Her passion is for developing novel methods and accessible software and tools to interpret big biological data in order to answer research questions more efficiently and more accurately. Her collaborative ethos instills best practice statistical analysis beyond her own research to ensure better decision-making nationally and internationally. Kim-Anh is strongly committed to statistical training and education to empower researchers from diverse backgrounds and to increase the capacity of the next generation of expert leaders in both Australia and worldwide. Kim-Anh is an expert in multivariate statistical methods and develops methods for omics data integration. And today she'll talk about her R toolkit, mixOmics, that she's been developing since 2009 to help researchers mine and make sense of biological data. So over to Kim-Anh.
Thank you, Christina. Yes, it's a great pleasure for me to present you about the methods that we have developed in mixOmics. And those methods are, were developed specifically to integrate different omics data types. So this is what I'm going to talk to you about today. What do we mean by data integration? And then how do we reduce the dimension of those data sets? How the software is organized, and then I will give you a few key examples of data integration analysis we have conducted in my team, and I will talk a little bit about the extensions that we have made in this package. So this is the field where I work. I work at the interface between biology and statistics, and my job is really to try to understand the biological question and translate it in statistical terms so that we can develop appropriate methods that can answer those questions. And so a close collaboration is needed between different fields, between statisticians, bioinformaticians, and molecular biologists. And that's because we have a very large amount of data that we can generate now, and they can come from different technological platforms and different sources too. That means that we have computational issues that we have to anticipate, and we also have to anticipate with the artifacts that come from those technologies and be aware of those. And most importantly, because those data sets are very complex, usually they have a small number of samples, it's important to rely also on the biological interpretation of the results. And I will show you how the tools that we have developed really rely on the biological interpretation. So we have evolved in past years. We have made tremendous advances in technologies, and that means that we have shifted the view of how we see a biological system. We went from a reductionist type of approach where we looked at one gene, we would do an experiment on one gene and then perform a statistical test on that gene, to a holistic approach where we can measure thousands of molecules at once. And the question about which molecule we should focus on is not very clear. And so this is where the challenges come from in systems biology. So we know that there are different omics that we can measure, we can monitor using those technologies. We can measure transcriptomics, proteomics, metabolomics. We know also that there is a biological dogma underlying those different omics. And so we can go from translation to expression to function. We can also measure other types of omics, for example, the microbiome, so the study of all the microorganisms in a specific ecosystem. And also there are other types of data types that we can also have access to, such as clinical data. But the problem is that, in fact, the relationship between all these omics data is not linear and it's not straightforward. We know, for example, that transcription factors have an effect on the transcripts, for example. And so looking at those data types altogether is challenging. So earlier before we had we would adopt a conventional approach where we would look at a specific gene or specific transcripts and study them in a reductionist type of approach. When we had more advances in the high throughput technologies, then we started to look at single omics and looking at, you know, a specific type of molecules, for example, transcripts, and then understand what is going on with those transcripts. So it's we adopted then a hypothesis-free approach because we didn't have a very clear question or a very clear hypothesis about which transcript to look at. And now we entering the arena of transomics or multi-omics, where we measure several thousands of molecules from different omics platforms in order to obtain a holistic view of a system. But that doesn't mean that we have to completely, you know, forget about a reductionist type of approach, because what really we're trying to do here is to try to identify the key molecules that are going to tell us more about the biological system and go back into a reductionist type of approach to generate new hypotheses. There are different ways that can define data integration, and actually the definition itself is quite blurry. So imagine you're doing a parallel assay. You can do a proteomic assay or you can do a lipidomic assay. Some people may want to analyze each data set individually and then try to integrate the statistical results. But there are other definitions where the data integration happens at different levels in this diagram. For example, at the bioinformatics level or at the analysis level. So I'm talking about data integration at the analysis level. And there are different types. The simple one is a multi-step approach where you would look at analyzing each omic data set individually and then somehow combine the results. There are other methods that are looking at the correlation structure between all these different omics molecules. You have also Bayesian approaches. My focus in mixomics is to work with matrices and specifically looking at matrix factorization to extract the common information from those different data sets. And so really what we're trying to do here is saying that because those molecules act together in a cell, we need to model them appropriately and identify them using novel statistical tools. So what we're trying to do is to shift the one gene hypothesis paradigm in order to obtain deeper insight into biological systems. And to do this, we use multivariate analysis, where we're going to identify a combination of biomarkers rather than univariate biomarkers. We are also going to reduce the dimension of the data, and that's going to help us to understand the complexity of those biological systems. And we will be able to integrate these different sources of data. And just as a disclaimer, this is a very famous quote from George E.P. Box, who is a statistician, and he said essentially all models are wrong, but some are useful. It has to be put into context. What he's saying is that we are trying to approximate the biology using statistical models, and we need to take that into account in our interpretation. So what are the expectations from omics data integration? There's several of them. You may, if you do a multi-omics experiment, you may want to have an overview and an understanding of what is the role of each omic to describe your biological system. You may also want to understand what is the relationship between all these different omics data types. You may want to identify a molecular signature, so that is more to explain the molecular mechanisms of a biological system. You may also want to predict. So based on all these omics data types, you may want to then predict whether an individual is going to have a disease or no disease based on what you have learned before on those data. And often the question I have the most is actually that you would like to do all of this at once. And so the method I'm going to talk about today is how we can integrate those different types of information, reduce the dimension of the data in a multivariate way, and identify those molecular signatures. So I'm going to give you a brief overview of what I mean by dimension reduction and how we do this. So we use matrix factorization methods, and basically what they do is that they build what we call components. Components are artificial variables that aggregate the variables that we have observed. So here in my case, variables are the genes, transcripts, the proteins, and we are going to aggregate those variables in order to summarize the sources of variation in the data. And for us, the variation in the data is the information from the data. And by doing that, we're going to reduce the data dimension. They are non-parametric methods, which means that we can handle difficult data characteristics of some of the data, such as a lot of zeros or high variability. And we are going to capture and explain the variation from the data. So very well-known methods, multivariate methods, are principal component analysis. But we also will focus on projection to latent structure methods called PLS, because those methods are very useful for data integration. So how do we decompose a matrix? So let's assume we have a transcriptomics data set. I have N samples in rows and P features or P genes in columns. So this is my expression data matrix. This data set is very big, and so what I want to reduce this data set. So I can reduce it by saying that actually I can decompose my matrix into a set of component scores and a set of loading vectors. And basically, if I can multiply those two types of vectors, I should be able to go back into my original matrix. So this is really what principal component analysis is doing. So you have those component scores, and so here I have two scores, for example, or two components. And the element in each component is actually a linear combination of my original variables. In order to calculate this linear combination, I need to know how I can weight those genes. And so those weights are indicated in the loading vectors. So if you have access to the loading vectors, you can calculate your component scores by multiplying each weight of each variable with the expression of those variables, and then you can obtain your components. What is important here is that we will obtain a small number of components, and they should be enough to summarize the information from the data. So we go from a data set that is of size P, the dimension, to a data set of size maybe 2 or 3. There are other variants of PCA, and one we've been working on is PLS discriminant analysis. So this is different from PCA because this time we said that we have an outcome. We have an extra information about the samples. So we can say, for example, the disease stages of the samples. And so we also obtain component scores, but this time we tell the method that we want to actually discriminate as best as we can our sample groups, which means that the loading vectors that we have here are going to change so that we can discriminate those sample groups. And so we are going to put more weight into the variables that are discriminative and can help to separate those sample groups. So here I'm not talking about any statistical criteria that we use in order to find those loading vectors and scores and component scores, but of course they exist. We either trying to maximize the variance of the components, or we're trying to maximize the covariance between the outcome here and the data set here. The other variant we worked on recently is the sparse PLSDA. And so the idea of the sparse PLSDA is that instead of taking into account all the genes in the data set, we're going to penalize some of the weights associated to the genes, so that only the important variables or the important genes are going to be assigned to a non-zero weight. And that means that only those genes that are represented in gray here are going to be used to define your component. And any of the genes that are flagged in red here are not going to be taken into account in the calculation of the components. So that means that based on those loading vectors, I'm able to say that, for example, there are 4 genes here that are important to define my first component. And that first component is trying to explain the difference between my sample groups. Then there are some other 6 genes here that are important to define my second component. And that second component also helps to further discriminate my sample groups. Now, I talked about dimension reduction. And so basically what we're doing is that instead of considering those big data sets, we only consider the component scores. And you can plot them. So this is an example here of a sample plot from PCA. So we have component 1 versus component 2, and the dots here represent the different samples. And what we're looking for in PCA is basically how similar my samples are, but it is an unsupervised analysis in the sense that we don't know what is the groups of those different samples. In discriminant analysis, it's different, as I show you, because in the method we include the information about the sample groups, and so we specifically ask the method, can you find the components so that I can discriminate as best as I can those sample groups? So my samples here are now in different colors because I know the sample groups. And so here we're doing a supervised analysis. And so you plot component 1 versus component 2, and you can visualize a big data set into a 2-dimension here, for example. And then you can go further than that. And so that's what we've been working on in the past few years, is to say, okay, now we know how to discriminate, sample groups in one data set. Can we now integrate all these different types of information? And so the idea is the same. We have now different data sets. You may have transcriptomics, you may have proteomics or metabolomics. They're measured on the same samples. We do this matrix decomposition, but this time we said that we want to maximize the correlation between the components. And remember that those components are linear combination of each of those data sets. For example, I have a component scores that are a linear combination of the genes, and then I have another set of components that is a linear combination of proteins, and so on. But the way I calculate those components are with my loading weights, and we define those loading weights in order so that the correlation between those components is maximized. And this is really how we achieve data integration here. Basically, we're asking, can we identify the optimal combination of these different omics variables, so that in the end, my components are going to be highly correlated, which means that they are going to explain similar information. And again, you can then extract those component scores and you can plot them. And this is showing you an example where we have 4 different omics data types, mRNA, microRNA, methylation, and proteins. The colors here represent the different sample groups. Here, this is in breast cancer. And here you have component 1 versus component 2 from each of those data sets that was extracted with our method. And you can see that you can see the similarities between samples, but you can also see that between all these different data sets, all these different omics data sets, we have been able to extract similar information. So for example, here the basal samples are clustered together, and it's the same in the methylation data or the proteomics data. But what you can see is that sometimes some data sets are not so good at discriminating a sample group. So for example, the microRNA, maybe the discrimination between the basal subtypes and the other samples is a bit more blurry. You can also have a consensus plot that's going to show you basically the average between all these different component plots into one single plot. We have also worked, so I told you that we can also identify a molecular signature. And so we've also worked on how can we visualize those multi-omics signature. So one way is to do a heat map, but instead of having samples and variables, here you have variables and variables. And that helps us to visualize the correlation structure between those variables that were selected. So here we're looking at, for example, cells and genes and metabolites. We also have developed circus plots, and basically you have the different variables here are represented on the external quadrant, and the color here represents a specific data type and a link between a specific variable that was selected with the method with another variable will represent the degree of correlation between those variables. So we really focus on how correlated our signature is, and then how discriminative our signature is also. And just one note, usually in multivariate analysis, we don't talk too much about p-values because, as I showed you before, we're trying to shift the univariate paradigm. So if we do have a p-value, it would be more to describe, how far our result is from random, but you can't really assign a p-value to each of those molecules individually because we consider those molecules as a whole. We aggregate them in a linear combination. So we're not looking at specifically the contribution of a specific molecule to describe a biological system. All right, so I'm going to talk a little bit about the package, mixOmics. So it's an R toolkit, and as I showed you before, it's based on these matrix factorization methods, and we specifically focus on feature selection or variable selection and visualization. So you may have different questions. You may want to be interested in analyzing one data set or integrating two data sets or more than two data sets. And so for each of those questions, you can apply different types of methods. And for most of those methods, we have developed a variable selection procedure inside so that we can identify those molecular signature. And what is good is that because they are matrix factorization techniques, we have access to those components, and then we can use those components to visualize either the sample. So I'll show you a few sample plots, but we have more than that. And we use the loading vectors as well in order to represent the correlation structure between those variables. But it's more than an R package because we focus a lot on methods development and also their application in the omics fields. So this is more like a project that is spreading in Australia, France, and Canada. We provide a lot of tutorials because we're very focused on education and training. So we run a lot of workshops. We've been running them since 2014. We run one-day workshop to three-day workshops because our aim is really to build a strong user community. So you can have a look on our website. We advertise those workshops on our website. And there are in total 19 methods that have been implemented. 13 were developed by my team. And as I said before, it really depends on the type of question you have. So for example, if you have one data set and you want to do a supervised analysis, you may go down that path and select that method. But maybe you are interested in integrating different data sets, maybe and not in a supervised way. And so you will have access to other types of methods. It's very important to understand that each method is going to answer a specific biological question. So I'm going to focus more on data integration, and we have two definitions of data integration. The first one is what we call the N integration. So in N integration, we consider different data sets or different omics data sets that are measured on the same N samples, but we have different omics. You can also have an outcome if you're interested in doing a supervised analysis. We also developed a method for P integration, and that's different. It's when you have the same omic data type, so for example, transcriptomics, that is measured across different labs or different studies. We have the same number of the same P features, but the samples are different, and we're trying to combine those data sets together. And in both cases, the problems are the same. We have usually a small number of samples and a very large number of variables. We have a variation that is not wanted. It's due to the differences between the omics platforms or between the different labs or the different protocols that we have used. And so our methods are really trying to focus on what is the common information between all those data sets. All right, so I'm going to give you 3 examples. 2 are focusing on N integration and 1 is focusing on P integration. And I've been selecting those examples to show you the diversity of data sets, data types that you can analyze with mixOmics. So the first one is called the Small Big Study, where we looked at the first week of human life. So it's a very exciting study. It's a small study because we could focus on a very small amount of biosample, so less than 1 ml of blood in newborns. And we could extract those at 2 different time points for a given newborn. So at day 0 when they're born, and then at day 1, day 3, or day 7. And from there, we could have then we could measure the transcriptomics, the proteomics, the metabolomics, some immunophenotyping measures, and some other types of measures. What is great about this study is we had a training data set from Gambia, but we also had a validation data set, and that was really important for us to validate our results from our analysis. So if you do, what you should do with a multi-omics study is to first start analyzing each single omic separately in order to understand what's going on in each of those data types. So this shows you the number of differentially expressed transcripts between day 0 and day 1, or day 0, day 3, or day 0, day 7. And those numbers here represent the number of transcripts that are either downregulated or upregulated. So you can see that with time, there's changes that emerge, whether you look at the transcriptomics, the proteomics, or the metabolomics. Actually, the first day of life, the first week of life, the molecular changes that happen are not random. They seem to be very ordered. So the second step then is to say, well, let's have a look at the correlation between those transcripts, or the correlation between those proteomics, and combine them all together. The problem when you do this and you represent them into co-expression networks is you have those big hairballs, and it's actually very difficult to extract any meaningful information from this. So instead of doing it naively, we thought, well, we may as well use the DIABLO, the N integration method, and see if we can actually identify a more structured correlation between those molecules. And so this is a sort of a heat map once we have applied DIABLO and identified a multi-omics signature, and you can see that there is some structure that appears. It's represented here in this simplified network. We were able to identify two types of subnetworks or modules, and each of those modules are based on those different data types and different molecules. DIABLO is a supervised method, so you can see this is a sample plot. So this time here we're looking at the samples, and you can see that we see a difference between day 0, day 1, day 3, and day 7. So what we found from this analysis is that we could identify new pathways and also common pathways. So new pathways that were identified only with this data integration method, as well as pathways that were common between all these different data types. So that really helped us to understand better the molecular mechanism underlying the first week of life in human. So if you want to see, if you want to know more, you can have a look at this paper. We have validated those results in the validation cohort. And to do this, we have done a lot of predictions. So basically, we asked, based on the molecular signature that we have identified in DIABLO, can we then predict in this new data set whether the sample is going to be a day 0 sample, or a day 1 sample, or a day 7 sample. And so the results validate really well. The other example that I'm going to show you is in single-cell transcriptomics. So as I told you before, those methods are non-parametric, which means that we can actually apply those methods to different data types. Single-cell transcriptomics data are characterized by a very large number of zeros, for example, and so we have using those multivariate methods are actually quite interesting here. So this is a benchmark data set that we have generated with my colleagues from WEHI, the Walter Elizabeth Medical Institute in Melbourne. And basically what we did is we had access to 3 very well characterized cell lines. They're represented in colors here. We divide them into single cell in about equal amounts, and then we sequence them using different isolation protocols, cell-seq, drop-seq, or 10x. And so basically we wanted to reproduce what may happen in real life, which is, you know, you have a colleague who did an experiment on those adenocarcinoma cell lines. Another colleague did a very similar experiment, but they used another protocol, and another colleague did the same. And then you meet them all and you say, hey, it would be great to combine all these data sets together, because individually I don't have enough cells here. I would like to increase the cells, my number of cells. So if you combine those 3 data sets together and you do you just do a PCA on this, the sample plot here shows the different cells, and the color represents the different protocols. And so what you can see is that from a PCA, it shows really clearly that the protocol effect is much greater than the cell line effect. So here you would like to see, we would like to see more a difference between the different cell types. So we have one type here, and then we have the 2 other types that are here. But instead, it's the protocol effect that that explains most of the variation in the data. And so here what we've done is we benchmarked different existing methods for single cell, and those are the different component plots that we obtained for each of those methods. We looked at how MINT and the MINT with the sparse selection is behaving. And basically what you would like to see in those plots is that the colors that correspond to the different protocols are all together. This is a good example where it's working really well, because we have it doesn't matter which isolation protocol was used, the cell types, the same the cells from the same cell type or the same cell line are together here, as indicated by the symbol. And so you can see that with MINT, it was doing an okay job, but we could still see a difference between the protocols. But once we had selected a subset of genes with MINT, then the overlap is much better. And so what it means is that really we were able to identify a gene signature across all these different data sets that were explaining the difference between the cell lines rather than the difference between the protocols. So this data set is available to the community. There are much more complex scenarios. You can test other different types of analysis, and you can find this data set in this paper here and also in CellBench on GitHub. The third example is looking at the host microbiota interaction in type 1 diabetes. So we don't really know what is the link or what are the functional consequences of the microbiome in the host. And so this is work with my colleagues from the University of Queensland in Brisbane. We looked at stool samples from the T1D prevention study. And using mass spectrometry, we were able to quantify the abundance of proteomics and the proteomics from the microbiome as well as the microbiome itself. So we have 3 data types, very non-invasive assays. And basically, we looked at 16S data, for those who know a bit about microbiome, and then the proteins from either the microbiome or from the host. And we had a cohort of 100 patients that were either healthy, seronegative, had a new onset of disease, or were seropositive. So we applied the DIABLO and integration method in order to identify what were the proteins from the microbiota, from the host, and what were also the microorganisms that seemed to explain the difference or seem to characterize those different sample groups here. So this is summarized into this kind of plot. It's an association network that has been generated on Cytoscape based on our results. And basically it shows a lot of information. The nodes here represent the different molecules that were selected with DIABLO. So for example, this is a human protein. This is a macroorganism, this is another this is a macroorganism. This is a macroorganism from a protein from the host. Sorry. And then the link between them represent the correlation. And then the box plot here shows the difference in abundance or expression for a specific molecule. So based on this kind of analysis, we were able to identify connected hubs of these different molecules. And those hubs here actually represent specific pathways that are very relevant for type 1 diabetes. So this is just to give you an example that now we are entering a new world where we can measure not only the omics from the host, but we can also measure the omics from the microbiome. And that means that it's opening new challenges in terms of data integration. So I'm going to discuss now just a few different extensions we've done with those methods. And one is that you can actually perform a pathway-based analysis. So what I showed you before were data-driven approaches. You know, we don't really know about the pathways of those molecules. We looked at it afterwards. After we finished the analysis, then we look at the pathway and we see whether it makes sense. But you can actually have a hybrid method where you include the pathway information in the analysis itself. So this is what we've done in an asthma study. We had 14 subjects and they had the allergen inhalation challenge, and the outcome, or what we looked, were the pre versus post challenge. In this study, we measured mRNA. We have 15,000 transcripts, different cell types, frequencies, and also some metabolite abundance. And this is the outcome here. So basically, based on this, we want to be able to discriminate pre versus post challenge to understand what's going on during the asthma allergen challenge. So you can do a pathway-based analysis. It's what we call a module-based approach. So basically, instead of looking at the mRNA, we had a lot of transcripts. We regrouped them into gene pathways. So those are regrouped in terms of non-gene pathways. And the way we did that is that for a given pathway, we calculated a component. So we calculated a linear combination of those transcripts that belong to that pathway. And so this is how we simplify a little bit the problem here, because instead of having, those two data sets, the metabolites and the mRNA, we work with gene pathways directly. So we have gene pathways and metabolites, and then we want to explain the difference between pre and post challenge. The other thing you can do also is to take into account the design. So we have a pre-post design in this study. And so if you do a sample plot with DIABLO and you look at the samples, what we see usually is that the difference within a given individual is very small. So if you have, this is the same individual, pre and post, and they are all linked between pre and post. And so you can see that within an individual, those two measures, pre and post, are very similar. And so it's difficult to disentangle the pre-post difference from the individual differences. And so in order to account for these repeated measures experiment, we do a variance decomposition. It's called a multi-level approach in mixOmics. And basically, it removes the individual variability so that now we can discriminate the pre versus post much in a clearer way. So we remove this variation that is not important for us. So this is what we've done in that study, and that requires another data transformation step, but it also helps you to maximize the statistical power. We've used that approach also in the small big study, and it was really powerful to differentiate day 0 from the other days in the neonate study. The other extensions that we're now looking at is how do we then go to a time course omics. So this is very challenging. So imagine that you have a multi-omics assay but you do that at different time points. So for example, here at time 1, you measure, the RNA, the metabolites, and the microbiome of some individuals. And then at time 2, you do the same, but maybe sometimes you are missing one data type. Sometimes you are missing specific variables in one assay. Sometimes you're missing some specific individuals, at specific time point. So there's a lot of challenges. We have irregular sampling, missing time points. We have usually a very small number of time points, between 5 and 10 maximum. The other issue we face is the high individual variability. At a given time point, you may see the variability of those individuals is quite large. The other challenge is that we are dealing now with different omics, so we're dealing with sort of 3D data structure. And also what we've noticed, especially in microbiome, is that the microbiome data are very prone to batch effects, and they're very sensitive to batch effects. And so that needs also to be corrected for. So the methods we're working on at the moment is trying to summarize the information of a given molecule. So this is an RNA molecule, for example, across time and across all these individuals. So the little dots here represent the individual values of each individual across time. And how can we, fit those, or how can we model those time trajectories? And how can we do this for every data type? And then What we're interested in is how can we then regroup those different trajectories according to their shapes or according to how they behave across time, irrespective of the data type, because what we really want is to identify functionally related molecules and see whether it can give us more light into those molecular mechanisms. So I show you an example here. It's the infant gut microbiota development. Basically, we were interested in identifying a bacterial or microbial signature. So this is microbiome data that would describe the dynamics of the gut microbial gut development in the first 100 days of life. And so you can see those different trajectories that we have modeled and then clustered using the method in mixomics. So the time is in the x-axis, and this is the abundance. And so you can see that for babies that were born through vaginal birth, we can see different shapes of those microorganisms across time. But when we looked at the babies that were born with C-section, we see very different shapes, and they correspond to different bacteria or microorganisms. So this is still work in progress. You can have a look at our GitHub code called mixOmics, but it's basically based on spline modeling and clustering based on the multivariate methods that I showed you before. And so those are my last slides. So if you're interested in mixomics and if you want to get started, the first thing is to install mixomics from Bioconductor. We did a transfer from Arcron to Bioconductor about a year ago. We have a book down vignette that is also hosted on GitHub to give you some pointers about how to get a handle of the different functions. We also have more detailed tutorials on our website. We have a forum discussion if you have any questions about the analysis, or if there are, any new options that you would like to see. You can say that to us. You can tell us if there's a bug. If you want to have more thorough training on how to use our methods, we run a lot of workshops at different levels and different topics. We're also very interested in training our trainers. So often they are participants that were there in their first workshop, and then eventually they are actually also training with us for the other workshops. And we also welcome a lot of researchers for scientific visits in our lab. Sometimes they come with their own data that they want to analyze. Sometimes they have a method that they're really interested in implementing in mixomics. And so to conclude, I hope I could show you that, those multi-omics studies are complex to analyze, and they require data-driven approaches to make sense of those data sets, and that we need to go from to a holistic and hypothesis generating approach. That doesn't mean that it should be data-driven only, and I'll show you that you can also include pathway information in the methods. But basically, we are at an exciting time where we work together between biologically focused and computationally focused scientists. And I hope that I could show you with those examples that the development of those tools is actually really important to make sense of those big data sets and to advance knowledge. And so those are the different people that I would like to thank. There's the mixomics team, which is spread between France, Australia, and Canada. We have also a lot of users that help us during the development process of mixomics. And there's also my team that was in Brisbane and is now in Melbourne, and a lot of collaborators who bring very interesting problems for us to solve. Thank you.
Thanks so much, Kim-Anh, for a comprehensive overview. I'll invite anyone now to put in their questions into the question box if you have any queries. We've got a couple in there that Kim-Anh's agreed to answer for us on the spot. Firstly, can omics integration methods also be used with categorical data, for example, genotypes, as in the integration of genomic and transcriptomic data in a multivariate fashion?
Yes, so that's a very good question that I often have. So there's different issues or challenges with this. The first one is if you are interested in identifying a signature, genotype data are probably not enough. You need to have, because they all have a small effect, those loci. And so the advantage is trying to combine all these loci together in order to explain a specific outcome. But if you are looking at variable selection specifically, it might not be very powerful. That's my experience. It depends, on the power of your study and so on. You can use genotype data in those models, but you're simply assuming that you're doing an additive genetic model. So you code your SNPs into 0, 1, 2, and you just assume it's an additive model. It can work, but as I said, I'm not sure they're actually bringing a lot to the table. The other option you can do is you can calculate a polygenic score on another study and include those instead of using the genotype data directly, and then use those polygenic score as a variable that you integrate with the rest. That might work. We are still working on other methods to deal with genotype data more efficiently currently. So it's still work in progress.
Thanks. We've got another question. I'd like to know more about what's needed before I start data integration, particularly how you normalized and transform each data set.
Yes, also a very good question. Because our tools are applicable to any omics data type, we don't specifically focus on normalization because, as you know, every platform is different. And so every omic data type generated from a specific technology will require a specific normalization method. That's why we don't even want to go there. So it will be your job to normalize the data accordingly, according to what is the consensus in your field. The only exception to the rule is for microbiome, where we thought we would just give a little bit of help. And so for microbiome, you start from your count data, whether from Shime or it depends, which software you use. And you will see on our website, we give you some information on how you go from count data to proportional abundance, and then how you transform them in order to analyze them in mixomics. So basically, we assume that when you are ready to use mixomics, your data are clean, they are normalized, they have been filtered. If you any variables that is too noisy may have been filtered if you want to. You don't have to, but that helps. And then the next steps for me are to analyze each data set individually first, as I showed you before. Is do a PCA or do a PLSDA on one data set at a time. Make sure you understand the sources of variation in the data, understand how complex your data sets are going to be before you go to the integration. I would definitely would not recommend you integrate the data at once first, because there's so many things that can go wrong in the process if you do not understand your data perfectly.
We have another question here too, more of a general question. In the design of a mixomics study, what things should be considered for a smooth analysis?
Yeah, so as I said before, understanding each omic is important. the experimental design matters a lot. And so you need to make sure that, you know, you've done everything you could to not have any batch effect, for example. You know, if you have a different sequencing run that you need to use, have you randomly sampled your samples on each run so that you don't have a run effect, but you can actually see a difference? Also think that sometimes people want to do too many multi-omics at the expense of having not enough samples. And, you know, the sample size is still important, even though, you know, we are exploring the data here. Sometimes there are omics that are not useful to describe your biological system. So I would say in order to have a smooth analysis, you really need to think about your design first and making sure that it's been designed so that it can actually answer your question.
Okay, well, thanks so much for answering those questions. That's all we've got time for today. So I'll just once again, thank you very much, Kim-Anh, for your time and fantastic talk today. DIABLO would like to acknowledge funding from Bioplatforms Australia and the University of Melbourne. And the ARDC would also like to acknowledge funding from NCRIS. Thanks once again and goodbye.
Thank you, and thanks for having me too.